So good morning. I'm Paola Bandini. I'm Associate Professor Geotechnical Engineering at NMSU, and I would like to welcome all of you to the Track 2 session on Biogeotechnologies for ground improvement applications. So the research and technologies that are being described, that will be described in this session, are sponsored and funded by the Center for Biomediated Bioinspired Geotechnics, or CBBG. The center is funded by the National Science Foundation and is a partnership of 4 universities, Arizona State University as the lead, New Mexico State University, Georgia Tech, and the University of California, Davis. And so the projects that will be highlighted today are projects that develop the biogeotechnologies within the center. So the first speaker is Dr. Ed Kavazanjan. He's a Regents Professor and Ira A. Fulton Professor of Geotechnical Engineering in the School of Sustainable Engineering and the Built Environment at Arizona State University. He's the Director of the Center for Biomediated and Bioinspired Geotechnics. Professor Kavazanjan is a member of the National Academy of Engineering and is a distinguished member of the ASE, the American Society of Civil Engineers. So Professor Kavazanjan, the floor is yours.
Thank you, Paula. Thank you for that kind introduction. And thank you for this opportunity to describe some of the research we're doing on mitigating fugitive dust using this process we call EICP, or enzyme-induced carbonate precipitation. I thought I'd give you just a quick overview of our journey today, since some of it in the beginning might seem a little off topic, but I'm going to start talking a little bit about natural carbonate precipitation processes, then our engineered carbonate precipitation process that we call enzyme-induced carbonate precipitation, or EICP. I'll talk a little bit about the importance of fugitive dust control, although I suspect I'm preaching to the converted in this session. Describe some of the laboratory testing work we've been doing, talk about the life cycle sustainability assessment work that we do to support our technologies, and then our upcoming plans for field testing this technology. And then I'd be happy to answer any questions that arise in the time that's allotted to us. So carbonate precipitation is one of the most common mineral precipitation phenomena in nature, probably right up there with salt. And the most common carbonate is calcium carbonate, calcite, or other polymorphs of calcium carbonate. It's the most studied biogeotechnical mechanism. Based on the number of papers that I get asked to review, it's really been an explosion in research in this area in geotechnical engineering, because if you can precipitate calcite, you can increase the strength, the stiffness, and the dilatancy of the soil. And there are many calcium carbonate precipitations, 6 or 7 natural precipitations we can identify, and then some man-made precipitations. So for instance, we have the abalone shell here in the upper right, which the material scientists tell you is really a miracle of nature. It's more resilient than any man-made ceramic. We have the white cliffs of Dover, which at least from an engineering perspective are remarkably resistant to erosion. And then here we have an anthropogenic mechanism. We have pipes where the carbonate mineral scale has developed on the inside of these wastewater pipes. So the process we're going to talk about, or I'm going to talk about, is EICP, engineered induced carbonate precipitation, where we use a chemical process called hydrolysis of urea, or ureolysis in short. And basically we put urea, calcium chloride, salt, an enzyme called urease in solution with water. We mix it all together. The enzyme hydrolyzes the urea. It breaks urea into a carbonate ion and an ammonium ion. Because we have calcium in solution from the calcium chloride, it combines with the carbonate ion and it precipitates as calcium carbonate. The ammonium also combines with the chloride to form, or the ammonium ion combines with the chloride to form ammonium chloride, which is a byproduct of the reaction that I'll talk a little bit about, because in high concentrations, it's a groundwater pollutant. In low concentrations, it's a fertilizer. So some people may have heard of this alternative process to our EICP process called MICP, microbially induced carbonate precipitation. In MICP, microbes that have the enzyme urease in their structure are cultivated and used to catalyze the reaction. In EICP, we use free urease enzyme. We extract the enzyme from a relative of the soybean, and we put that into the solution rather than the microbes. The benefit of that is that the enzyme unit is much smaller than the microbe, so we can penetrate fine-grained soils. We can penetrate silts, and of course, silts and fine sands are the soils most susceptible to fugitive dust generation. But there are some other benefits too that we see with the enzyme. One is it appears to produce, at least in some circumstances, higher strength at lower carbonate content. So the graph over here on the right, these blue squares and circles are all results of shear strength, unconfined compression strength on the vertical axis, calcium carbonate content on the horizontal axis, achieved by MICP. In the blue circle here, or oval, is our EICP process. So with EICP, we can get shear strengths of up to 2 megapascal, 20 tons per square foot, with less than 1% carbonate, whereas with the MICP process, they often need as much as 12 to 14% carbonate. What does that mean? That means we can use less chemicals, makes it cheaper. It means we produce less ammonium byproduct, so it's easier to manage. And we're using this process for a variety of different purposes. I'm going to talk about fugitive dust control, but you'll also hear in this session from Paula about erosion control, and from Kimberly about making columns of biocemented soil using this process. A quick word on dust control. I think this is preaching to the converted, where we live here in the Southwest, but it's a significant air quality issue, and it's primarily due to wind-blown soil. It's not vehicle emissions, it's not industrial emissions, it's soil. And it plagues many arid, semi-arid areas around the world, not just the Southwest US, but Eastern China has some serious problems, North Africa, and other arid and semi-arid regions. And it's known to cause serious health problems. So it's a priority pollutant. And at least where I live in Maricopa County in Arizona, for a while we were threatened with loss of all our federal transportation funding because we didn't meet federal guidelines for fugitive dust. So how do we control dust? Well, the traditional dust control method, the most common one, is water, right? We run a water truck. Now, in Arizona, and I suspect in New Mexico too, when it's 110 degrees and 10% relative humidity in the summer, you can run that water truck until the cows come home. And as soon as it drives away, the soil dries out. So it has limited effectiveness in arid, hot climates. Our mining friends use salt solutions, typically magnesium chloride. But for civil engineering purposes, these are frowned upon because they're environmentally unfriendly. They can lead to groundwater and surface water pollution. We also have synthetic polymers, which work well, but they're very expensive. So for long-term dust control, people will go to these polymers, but for short and intermediate term, they're really not cost-effective solutions. So our concept, our biogeotechnical EICP fugitive dust control concept, is to create a wind erosion resistant carbonate crust by spraying our EICP solution onto the soil. And right now we use a sprayer, but I think it'd be easy to just put an attachment on a water truck and fill it with our solution and make one pass of the site with the water truck. And so in the picture here on the right, you see a typical crust that we created in the laboratory by spraying our EICP solution on a fine-grained soil. The advantages of the EICP method is it's one and done. You don't have to drive the water truck, from dawn till dusk, 6 days a week. You do it once, and it'll last at least until you disturb it again, until you ride over it again. But even then, it will still provide significant benefits in terms of mitigating fugitive dust. Although one of the issues that we've tried to assess in our life cycle cost assessment is at what point does it really become cost effective? It's a little bit higher initial cost, but you don't have to run a water truck continuously. So from a life cycle basis, it'll have a reduced cost. What are the disadvantages? Well, the initial cost, higher initial cost, and then the potential environmental impact of that pesky ammonium chloride that we generate. So we started our research in this by doing wind tunnel testing. And we're fortunate at ASU, we have a wind tunnel that was built to study wind and dust storms on Mars, the ASU NASA Planetary Wind Tunnel. And we had to retrofit it for our samples. This is a retrofit so we can put our sample in the bed of the wind tunnel. And we had these very sophisticated sample containers. They're 9-inch pie plates, pie pans from Costco. Here is one of our graduate students preparing one of the specimens. The black you see is just a shroud to catch the overspray. He's got the cementation solution in this little hand sprayer, and he's spraying on a controlled amount of solution over the surface of the soil prior to loading it into the wind tunnel. And again, here's one of those specimens that's been dissected. You can see the cemented crust that formed, and then you can see the uncemented material. This was a natural silty sand from Arizona. So just to quickly summarize the results of the wind tunnel tests, one challenge you have with fugitive dust is there's no regulatory standard. What do you want to compare your engineered solution to? So we said, well, the regulatory standard is keeping the soil wet. It's thoroughly wetted soil. So we took our test soil. In this case, it was a fine sand, Ottawa F60 silica sand, and we thoroughly soaked it and then put it or wet it with the sprayer. Soaking is perhaps a little strong, and put it in a wind tunnel and measured what we call the threshold detachment velocity, the wind velocity at which soil particles start to detach from the surface and become ingrained in the airflow. And for this particular soil, the threshold detachment velocity was about 23 meters per second. That's right about 60 miles an hour. And dry soil was down here at about 10 meters per second. So then we treated it with 3 different EICP solutions, 3 different concentrations. We wanted to find what concentration gave us the same erosion resistance as water. So we started with a 0.2 molar solution, that's the chemical concentration, and we maxed out the wind tunnel. We couldn't tell you, we maxed out our wind tunnel at between 25 and 30 meters per second, and at the maximum velocity, we still had an intact crust. So we reduced it to a 0.1 molar solution, and we're pretty close. We're just a little bit more resistant than wetting it. We said, well, maybe we can save even more money by reducing the solution again. We got it down to 0.5 molar, but then there we didn't match up with the wetted soil. So we said, well, for this soil, the appropriate concentration is 0.1 molar. We also compared it to controls in which we used the salt, because remember, salt solutions are an accepted method of dust control, but we didn't put the urease enzyme in. So there was no hydrolysis, no carbonate precipitation. And so these are the same 3 concentrations of our control specimens, the salt solutions, calcium chloride solutions, and even at the 0.2 molar concentration, it didn't quite, and this was after they dried, of course. We sprayed them with the salt solution and then let them dry. But even then, the salt crust wasn't quite as resistant as the thoroughly wetted soil, or certainly as our EICP treated soil. We support all of our research at the center with life cycle cost assessment. Whoops, I went one slide too far. And the first thing we did was what we call a hotspot life cycle sustainability assessment. Not just life cycle cost, but we look at life cycle environmental impacts and life cycle social impacts. And the results of this analysis gave us insight into the things that govern the commercial and environmental viability of our product. We found out that the cost was dominated by the cost of the urease enzyme. Now, we were buying our enzyme from lab supplies, so we were paying a couple $100 for a little bottle of enzyme that was lab grade, suitable for pharmaceutical purposes and for food purposes. The energy impacts were dominated by the energy that goes into urea, because most urea is made from natural gas. The environmental impacts were dominated by the ammonium chloride byproduct. So this hotspot analysis told us that we needed to lower the cost of the urease to be commercially viable, that these green processes for producing ammonia and urea would be very beneficial should they come into play to reduce the energy impacts. And we needed to be conscious of this ammonium chloride impact. We needed to address that. So the first thing we turned our attention to was the low-cost urease. And as I say, our initial experiments used the lab-grade enzyme, very pure, very expensive. We didn't need something that pure for dust control. Now, urease is found in all green vegetation, and the richest source of urease is a relative of the soybean called the jack bean. In fact, we tried a bunch of different, we tried soybeans, we tried watermelon seeds, a bunch of different substances. And basically we learned that the pharmaceutical companies weren't stupid. The pharmaceutical companies use jack beans because they're the richest source of urease. And so we started working with the jack beans. And we developed what we call a crude extraction process, where we skipped the 3 purification steps used to make pharmaceutical grade enzyme, because we said we just don't need that purity. We replaced the extraction solution with tap water, and we found out that the resulting solution worked just as well, and in some cases even better than the pharmaceutical enzymes, that the impurities in there actually enhance the biocementation. And so now we have a process that just requires a supply of jack beans, a kitchen blender, and some cheesecloth. We can make our enzyme just in time at the job site, and it's reduced the cost of the urease by a factor of 40 times and made this process really cost effective. Now, as we moved on with our research, we went into a detailed life cycle sustainability assessment because we wanted to see at what point did the higher cost of the EICP method start to pay you back compared to water. And it's not only the cost of the water, but we're being better environmental stewards. We're using less potable water. There are less vehicle fuel consumption and emissions because we're not driving around a site constantly. We also had to pay more attention to the ammonium chloride because that can lead to eutrophication if it discharges into surface water body. The good thing is it also acts as a fertilizer. So if you're going to try and vegetate your site after you apply the dust control, you can have some benefits. It can be taken up by the vegetation. It can stimulate the vegetation. And there is a paper in the ASE Journal of Construction Engineering and Management that summarizes our life cycle sustainability assessment for this technology. Here are some of the summaries for that paper. In the paper, we assumed that the crust would last for at least 2 weeks. If we look at water consumption, or energy consumption, applying water and applying EICP, we're about 1/3 of the cost. When we look at water consumption, we used about 1/10 as much potable water. Lower CO2 generation, lower global warming potential, slightly higher acidification potential, but that's assuming there is a receiving body of water that could lead to eutrophication. Cheaper project costs. Once we took care of the fixed costs, we found out that the project costs were about the same after a week, and then a much lower social cost of the carbon that the process creates. So where are we now? Well, now we're moving on to do field tests. We have 2 test sections at Arizona landfills. A couple of months ago, we completed the short-term test where we worked out our procedures and processes for creating the crust. We have a long-term test that's now set up and ready to go at a local landfill called the Salt River landfill. We'll put down our spray this month, and it'll be in place for 6 months to a year with periodic measurements. And we just finished negotiating a contract with the Bureau of Reclamation to do a test section down at the Salton Sea, where they have a tremendous problem with dust control. The Salton Sea is a man-made sea that was created about 1910 when the Colorado River aqueduct breached and drained into a shallow depression, became a lake. Recreational activities grew up around the lake, but now they've cut off the supply, and so the lake is shrinking. And as it shrinks, it exposes mud flats that dry out and create these fugitive dust storms that are contaminated with herbicides and pesticides from agricultural runoff. And so the Bureau of Reclamation is under a consent decree to mitigate the fugitive dust. And we will be doing a test section there within the next 6 months. So that's a brief overview of our technology. With whatever time is left, I would be happy to answer any questions you might have.
Okay. Thank you, Professor Cavazanjan. So I actually got a couple of questions from in a text. So they say, will it be required to train field personnel for this method?
Anybody who can drive a water truck, or anybody who can hold a hose in their hand, that's really the only training that's necessary. The field test sections, it's implemented by our graduate students. So We have a mixing tank on a trailer. We have two solutions that we mix together at the nozzle and we spray it on a site, just like watering your lawn with a garden hose.
Okay. And another question on how large are the test sections?
So the test sections, each test section will have 4 or 5 different plots where we'll try different concentrations. We'll have a control plot with no dust control, and we'll have a magnesium chloride control plot. And each plot is about 1,000 square meters. 1,000 square meters? I think that's correct. For the Salton Sea, for the local landfills, they're a little bit smaller. They're about 2,000 square feet each.
Right? Mm-hmm. And the last question I have here, it says, one just came here, does the prepared solution degrade or decompose over time? How long does it last in typical environment?
So the challenge with the solution is that the precipitation occurs very quickly. So that's why we have 2 separate containers. We don't mix it till it gets to the nozzle. If we mix it all in a tank, it'll start to precipitate in the tank. It's a very fast reaction. If we don't mix the 2 solutions together, typically we have a calcium chloride solution in one tank and we have the enzyme solution in another tank. If we don't mix them together, the calcium chloride solution is good indefinitely. The enzyme solution, if you leave it out in the sun over the period of a week, it might lose about 30% of what we call its activity, which describes how strong the urease is. So over a period of time, that would start to degrade. But there's no reason to, you can just mix it just in time.
And the last question, before we move to the next presentation, can you comment on the duration of the treated surface?
So we've actually done accelerated erosion testing. We have a paper coming up in the next International Soil Mechanics Conference where we've put our EICP treated specimens in the accelerated erosion testing device that's typically used to test asphalt pavements. And what we find out was that the heat and the ultraviolet radiation actually make the crust stronger. It actually cures our carbonate crust. So unless you disturb it, unless you roll over it with a piece of equipment and start to break up the crust, as far as we can tell, it's going to be very durable, very long lasting. If you had acid rain over a period of a year or so, it might have some negative impact. But the pH really has to drop below 5 before you start to dissolve some of the carbonate. So we expect it to be very durable, but that's why we're doing the field testing.
Okay, yes, thank you. There is one more question in the chat, but you can answer and everybody can see the question as well.
Okay,
right?
I'll go there.
Okay, so Anne, release the screen. Our next presenter, it's my pleasure to introduce the next presenter, Dr. Ferran Garcia-Pichel. He is a Regents Professor of Microbial Ecology at Arizona State University, working on environmental issues. He is the founding director of the Center for Fundamental and Applied Microbiomics at ASU. Professor Garcia-Pichel is a senior investigator in the Center for Biomediated Bioinspired Geotechnics. And one of his research areas is developing strategies for the sustainable recovery of degraded arid lands. Professor Ferran Garcia. Yes.
Yeah. Good morning, everybody, and thank you, Paula, for the introduction. It is my pleasure today to tell you about a little different approach to the same end in this respect. So we, after many years of research on this system I'm going to talk to you about, we decided that it was high time that we would use it for some applications. And so I'm going to talk to you about natural biocrust microbial communities used to stabilize soils against erosion and the production of fugitive dust in open settings and then also in solar farms. And we will see why that is. Let me see. Okay. So just to acknowledge that the work I'm going to talk about is the work actually of many of students and postdocs and undergrads working in our lab. And so here are their names. So here's a natural soil. This is a southern Sonoran Desert, very similar in many cases. You would see that the vegetation, which typically is the major agent of dust control in temperate environments, is rather absent. Low concentration of plants, high open interspaces that may seem to many that are barren. However, when you look close in those interspaces, then what you find are these biological soil crusts. And in this picture, you can see that the soil in the interspace has been sampled, and you can see that it's sort of crusty. These are the communities of microbes that take on the role of fixing the soil under natural pristine soils in arid lands. So let's see, my things are not advancing. Okay. I think it's just being slow. So what is a biological soil crust? Just a little bit of a definition. It's a topsoil organosedimentary assemblage of microorganisms that is built by microbial phototrophs. Phototrophs are organisms that make a living out of light and CO2. So they're photosynthetic. Cyanobacteria are some of them, algae, lichens, mosses, and so on. They come in a variety of what we call microbiomes, a set of bacteria that live together with them in mutualistic interactions. Where do we find this biological soil crust? Well, anywhere where plant litter allows the absence of plant litter allows the light penetration to the topsoil, then you have a habitat in the topsoil for these organisms. And so typically we find them in arid lands, polar soils, but interesting for us, in arid lands, they're very common. Here's a picture that some of you may recognize in one of our field sites in Las Cruces. And then you can go into the plant interspaces and look at the crust and pick up the crust. So this is what we're talking about, a very hard shell of microorganisms. In fact, you can see some of the filament of microorganisms holding just under the crust some of the soil grains against gravity. You don't see the filaments, but you see the floating sand grains. These are our crusts. And you can see them after a monsoon rain, for example, in this picture also at Las Cruces, where you see the green of the chlorophyll that the microorganisms have to make photosynthesis be very patent, where you would actually just see a cryptic, apparently barren soil otherwise. That they have an impact on the protection of soil against erosion has been clear for a long time. I'm not going to dwell on that. But they have a problem, and that is that the crust cover is very sensitive to anthropogenic impacts. Mostly in the US Southwest, historically has been grazing, but also currently off-road driving, hiking, in areas around cities, construction, urban sprawl, agriculture, military training. All of these things can have negative impacts on compressive forces that destroy the crust. And that is the typical situation that we see now. Okay, very slow. We'll give it a second or two. This is the typical situation that we see now in areas around, for example, Phoenix. I'm not sure why it froze. Okay, so we have the problem is that the absence of crust in most heavily inhabited areas result in a lot of fugitive dust. Our solution to this problem is to restore the microbial crust with what we call microbial reforestation, just as in temperate climates you would have reforestation. And our approach is, by analogy, to establish what we call a microbial biocrust nursery as a way to producing inoculum to inoculate soils that are devoid of crust, so to allow the natural restoration of the natural way of desert soils to maintain themselves against erosive forces. Most of the microorganisms that are interesting to do this, the natural ones, are filamentous, and these are some microscopic structures that you can see. This is a filament in the upper left of the typical microbes that develop in our deserts. They come together into what we call bundles of ropes that you can see at high magnification below, and you can see them in their natural habitat in the upper right photograph, where you can see how they sort of weave this mesh that holds the sand particles together. During our previous studies, we have learned quite a bit about about the biology of these organisms. We know that we cannot grow them in a way that, say, for example, the biofuel energy grows the same kind of bacteria, because they hate being submerged in water. So we have to find alternative organisms. We know that just like us, they are subject to pandemics, predatory bacteria that kill sometimes the crust. And we know that they love shade. We also know that the organisms are not the same in different geographical areas. Different organisms are the dominant ones as being adapted to the local climate. So here's how we start with our current project within CBBG and a list of problem solved and major roadblocks for our technology. We know which microbes to use. We know how to culture them by now. We know how to monitor them in the field. We know how to acclimate them for life in the soil, but we have a problem with mimicking the climate of the sites because different organisms like different climatic conditions. We have a serious problem with scale up of this technology to attain our goals. I showed you a little bit about some problem with epidemics, bacteria that kill bacteria, a little bit of delivery, and then we also need to start working on partnership at the mid-scale. So I'm going to tell you about our latest research in this area. So the first thing to adapt to dealing with climate is to, if it's too hard to make the lab adapt to the climate, then we'll take the lab to the right climate. And for this, we developed the mobile briar-crust nursery facility, which is now up and running. The human pandemic prevented us from moving it yet, but we're really eager to do that and going to Las Cruces pretty soon, hopefully, so that we can do all the laboratory procedures needed right under the climate that the crust are going to need. I'm gonna have to do this maybe. Yeah, all right. The second improvement we did was to develop the fog irrigated soil substrate approach to go ahead and use those technologies to create artificial crust that can be used to inoculate the eventual sites. And so this has worked pretty well. The problem, though, is that our current power, as we can calculate it, is about we can produce inoculum to cover 5 hectares per year per person working full time. So if you just look at Maricopa County, in Maricopa County, the county where Phoenix is, there's about 100,000 hectares of degraded desert that are the source of our fugitive dust. So you can just do the calculation. We don't have 20,000 years to do that, nor can we employ 10,000 workers. So we really need to scale up, and this is one of our major roadblocks right now. Our solution, or what we hope will become our solution, is using solar farm as nurseries, because as you remember, these crusts grow very well under the shade. And so these are natural, if you will, greenhouses that are ready for us on many desert areas. And so I think partnerships in this area can work out very well and allow us to scale up. So we started a project where we could test our ability to produce this this technology and apply it to one of these solar farm settings. Now, I'm probably gonna bore you with all the details of the flow through, but essentially all of the things, all of the microbiological approaches and the production of the inoculum which are in blue boxes, we're already there, and we're now in a project. We have deployed different treatments into the site, local solar farm, and we're in the monitoring phase to determine the optimal approaches for that particular application. So we have two types of biological inoculum we have produced. In this case, the microbes themselves grown with typical microbiological approaches, but also whole artificial biocrust that we grow from little biocrust remnants that we find in the site. And you can see here in the left, for example, that the community composition of this crust of our grown inoculum is very similar. Each color is one species, and the relative proportion very similar to the natural biocrusts. And the artificially grown crust in the nursery using FIS do really match what you would expect in terms of strength, holding on to the soil particles, and typical biomass. So we were ready with that. We decided then to go and prepare all the inoculum and inoculate the field site with the different treatments in order to find optimal approaches, different cultures, artificial crust, adding some beneficial bacteria, not adding them, and so on. And we deployed our inoculum, I think February 2019, so that they would be there and had taken hold just before the growth monsoon season. These communities only grow when the soil is wet and there is rain. So let me show you some of our results. Here you have in the first graph to the left, you have the basic, where untreated outside of the plots. Not much growth, very low chlorophyll A, which is a measure of biomass. And then the treated in yellow, for example, you can see in the second plot that we inoculated. We pretty much double the concentration of chlorophyll at initium. And then it started looking pretty good. They started to grow. After that, we decided we'll sample again after the growth season. And lo and behold, what we found was not growth, but death. Essentially, the populations went down after that summer. The same is true, or the same happened to the other treatments with natural biocrust. They didn't really grow. And so we were a little bummed out, but apparently it was a case of bad timing, because the monsoon was actually failed to show up this year. So we have one of the driest, one of the hottest summers on record in the valley. And so perhaps we thought it is good that they didn't all die. And so we decided to continue the monitoring for a further year. And so you can see that in the graph to the left here, the crust started to recover clearly with the winter rains. We have essentially continued, we will be continued our monitoring, hopefully through the next monsoon, which will show up for us, and we'll be then also monitoring the stability of the different treatments towards erosion. So the work is a work in progress. And with that, I think I will be taking any questions if there's any time.
I have here in the text, there are several. How far is this technology away from the point that it can be implemented in large areas?
It's far. I think we can do implementation in particular areas. So if we look at areas, say a hectare, on the order of magnitude of hectares, we could do it right now. Notice that we actually didn't want to water those inocula because we want, eventually that will not be possible at the large scale. But at the small scale, we could supplement with water and so on. So I think the technology is ready at the small scale. We're thinking of collaborating with, say, construction areas locally for individual houses and so on, or housing projects. But eventually our goal is to deploy this in large scale.
Mm-hmm. Another question, how do you control overgrowth of the microbes when they are moved from the nursery to the site?
So what we do, I didn't show that, but we do what we call CSI approaches. So we monitor the DNA of the soil, and we do community composition by sequencing DNA. So we know who's there and how are they growing. And that's essentially the graph I showed you when I compared that what we grow is very similar to the natural crest. That's how we do that. And the monitoring also includes this sort of thing. We want the right things to grow and not the wrong microbes to grow in place, because some of the microbes may not be very good at holding the soil together. We don't want those to grow in there.
Mm-hmm. And then the last question I have here is, does rain affect or wash away the bacteria?
Mm-hmm. If we do our job right, no, they don't, or no, it doesn't. So one of the abilities of these microbes is that they move, they're motile, and they go up and down the soil. So when they're dry, they move 2 or 3 millimeters below the soil surface and hold on in that sort of refugium until water comes. And that's why you see the soil greening up when it rains, because the bacteria come back up to the surface. So if they take hold of the soil, they're pretty impervious, unless the whole soil is gone.
Okay. Thank you. Let me double check on here. Okay, so if anyone else have other questions, you can write them in the chat, and Dr. Garcia Pichal will can answer to you. Okay. Thank you. Thank you very much. Very interesting. Thank you. And yeah.
Paula and I are now switching hats.
Yes.
She's the next presenter. So it's my pleasure to introduce Professor Paula Bandini, the Wells Hatch Associate Professor of Civil Engineering at New Mexico State. She's the campus lead for the CBBG research, and she's the CBBG lead for the infrastructure construction thrust of the center. And she's going to talk to us about soil erosion control using EICP.
Yes. So thank you. So I will describe the research that is being conducted at NMSU to develop methods for water erosion control in sands using the enzyme-induced carbonate precipitation technique, or we call it EICP. So this project is part of the CBBG, or the center, and as the other 3 projects that are highlighted in this session. So the project is a collaboration between NMSU and ASU. The team in ASU was the one that developed and spearheaded this technique for ground improvement. So in my presentation, I'm going to just briefly just indicate the motivation for this research and the goal, the development of the method, and some of the lab testing that we have been performing and the results, mention some ongoing and future research, and I want to highlight a unique facility equipment that CBBG has developed, the large outdoor rainfall and infiltration simulator. I will acknowledge the team that has work on this project, and I will take some questions after that. So some of the common methods for erosion control that are used in practice include rolled erosion blankets, geosynthetics for temporary and permanent protection, sprayed on polymers, but those tend to be proprietary materials and tend to be more costly than some of the other options, and may require some recurrent maintenance. Hydroseeding and mulching and native grasses and plants can be effective erosion control methods. but they may require fertilizer, especially if they are used in soils and sandy soils that have very little organic material and very little nutrients for the plants to grow. So you have to provide those. And it may also require considerable irrigation in dry climates. Otherwise, the plants, the seeds will not germinate. So even fertilizers and irrigation are applied depending when the project is completed. Heavy rain events may happen before the germination of the seeds, washing away the seeds and establishing the vegetation very difficult. So there are some drawbacks on the existing methods and challenges, and especially in terms of sustainability, maintenance, and performance. So our project uses enzyme-induced carbonate precipitation, or EICP. For those that join our session after the first presentation, I'm going to describe what EICP is. It's a biogeotechnical process catalyzed by urease enzyme, in which calcium carbonate is precipitated from the hydrolysis of urea. It's an abiotic approach, so it's been developed for ground improvement applications, and no microbes are involved, but the method mimics what the bacteria does. So the goal of the project is to develop a sustainable method for erosion control in sands, in sandy slopes, using the EICP technique to cement and create a superficial layer of soil that provides protection against runoff erosion. At NMSU, our focus has been on developing methods that work on native sands, and we have been interested in trying to control the thickness of the layer that we can form, and demonstrate the method in field sites in the future. So major achievements that CBBG project have had, especially the ASU team, have allowed the advancement of this project. For example, the extraction of the urease enzyme from the jack beans with a simple, low-cost method. So the experimental work that our NMSU project conducted in the last 12 months included multiple testing of block specimens to compare many different conditions and variables, box specimens, and plots 2x4 in area. So we are scaling up our erosion testing. So our students conducted and constructed and calibrated a portable lab scale rainfall simulator that can be adapted in the future for field testing. We are using it for the erosion control at the bench scale. The testing included carbonate content determination, mineralogy study using the X-ray diffraction, determination of the mass loss during the erosion test, and indirect assessment of the cementation strength. So the first series of block tests was conducted in 2 native sands, and Ottawa sand as a control, because we know that the method works well in silica sand like Ottawa. Specimens were prepared with a new mixed compact method in which the 2 parts of the EICP solution are applied to the soil separately to prevent the early precipitation. We wanted to look at that option as well. So this test series was study the effect of reducing the volume of treatment solution in subsequent cycles. And this is important because it can help reduce actually the amount of materials and cost to form a cemented layer. So we learned that and confirmed that the crude extract ureas that using the method that was developed by our colleagues at ASU produces comparable results or even better than the costly commercial enzyme that was available before. And we also realized that we were going to need 2 or more cycles, treatment cycles, to create a cemented layer that was strong enough for erosion control. So the second test series only used crude extract, because that's just to focus on what it was going to be used eventually in the field, and we focus on the treatment application methods. So 5 methods were compared: percolation and mix and compact, with different sequence of application. I want to highlight the top graph. I don't know if you see my laser pointer. The top graph shows the percent mass loss from the erosion test, the first erosion test. And the blue bars indicate the one treatment, specimens that were treated only with one time, and the yellow ones are with 3 treatments. And you can see how much improvement in erosion resistance we had just with that. And so in the top graph, we want the shorter the bars, the better. In the lower graph, we have the strength of the cemented layers. So we want the taller bars, the better, the stronger. And so we learned from this that the 3 treatments were providing enough strength and resistance to erosion, at least at the scale that we were performing the test. And we also confirmed that the two-step mix compact method was working well, as well as percolation. I'm going to skip this part, and I want to talk about the erosion test on plots that are 2 by 4, 2 inches thick, and each of the plots were subjected to 3 intensity levels of rainfall in the laboratory, simulated. 20 minutes each, 10 days, and then again, the same series of tests 30 days after the last treatment. And let me advance one more. So these 3 specimens are shown here after the erosion testing. The one on the left was prepared by percolation of the treatment solution. And you can see some of the white material on the surface, which is precipitation of calcite, that may have clogged a little bit the surface, because these were actually 3 treatments. However, it created a pretty strong layer, and it was a thinner layer, but stronger. And you can see here in this graph the results, the soil loss for the percolation method provided the smaller soil loss, and so the greater resistance to erosion. And so these bars indicate the soil loss in tons per acre for the 3 rainfall intensities for each of the methods. And these are actually average values. And we can see that the first event led to the highest soil loss. We were surprised, but we then understood that in the first event, there could be some weaker, partially cemented particles on the surface that were washed away. And after those were mobilized, and the remaining exposed surface was much stronger and had higher resistance to erosion. So ongoing research at NMSU includes assessing erosion resistance as a function of treatment. So we are looking at different number of treatments, determine the optimal size and location of the samples that we need to take to determine calcium carbonate determination in which part of the layer, in which part of the specimen or the plot, to provide guidance in the future for QA/QC in field test and eventually implementation. Also, our students are designing and conducting and eventually we'll conduct a field erosion test. We are assessing the feasibility of a field site at a local flow control dam that shows signs of erosion and is in the process of being repaired. And continue the development of the data and a full project for a sustainability assessment of this method. I want to highlight the CBBG large outdoor rainfall and infiltration simulator, LORIS. The simulator meets and exceeds compliance with ASTM standards for testing of erosion control products. The simulator has 25 rain gauges and an array of sensors that measure infiltration of water into the soil, percolation and storage, and runoff to determine the loss of sediments induced by erosion. So the equipment allows a 40-foot long, 8-foot wide, 25-ton bed of soil, which is possible because a significant feature of the rainfall simulator is the landfill tipper used to elevate the soil test bed. So, and an angle of 30 degrees, up to 30 degrees. So it was modified, the tipper was modified from a tipper used to empty municipal solid waste trailers, transfer trailers at the landfill. So the tipper can incline up to 30 degrees. And the facility for the rainfall simulator includes water storage and recycled storage tanks. The facility includes a weather station and also runoff tank to collect all the sediments from the erosion for the analysis. So this is an extraordinary rainfall simulator and will be used soon to test EICP treated sands for erosion and of course other products as well. And the facility is a CBBG from the facility that is at Arizona State University in the Polycampus site. I want to acknowledge my NMSU students that perform all the testing that I described initially for this project. Each of them leads a different part of the project, and my NMSU collaborators, Professor Mitchell and Wazir. And I want to thank the ASU collaborators for sharing their experience on the JCP technique. And Eric Escodo is the ASU PhD student who has developed the CBBG large outdoor rainfall simulator for his doctoral dissertation. So I will be happy to take questions if we have time.
So we probably have time for 1 or 2 questions, but I don't see any questions. If you have a question, you can answer it in the chat box.
Yes, yes. Okay.
So I don't see any, so I'm going to turn it back to Paula because we are a little over and we want Kimberly time to present. So Paula, it's back to you.
Very good. So I stop sharing. Okay. There you go. And Dr. Martin, do you want to have your video on? Okay, very good. So our next speaker is Dr. Kimberly Martin. Dr. Martin is a senior engineer at Keller, where she focuses on innovation and sustainability. She recently joined Keller after completing her PhD at Arizona State University as part of the CBBG. With over 13 years in the geotechnical industry, Dr. Martin previously worked at ExxonMobil as a lead geotechnical engineer on development projects across the globe. The floor is yours.
Thank you, Dr. Bandini, and thank you all for being here. So I'm taking this EICP technology that both Dr. Kavazianjian and Dr. Bandini spoke about and taking it deeper into the ground as a ground improvement technology, using it for biocemented soil columns. Okay, so just a quick refresher, EICP is the urea, the calcium chloride, and the urease enzyme put into water and injected into the soil, where we get calcium carbonate as our binder. That's what we're referring to when we say biocementation. So why we wanted to make biocemented soil columns for ground improvement is soil columns are a known way to do ground improvement for the transportation industry. They're often used to support embankments that are on problematic soils. And so in that case, it would do something like increase the soil stiffness to prevent excessive settlement, increase strength to increase bearing capacity. It could be used to prevent triggering of liquefaction, and also to provide cementation to support excavations in sand. So in transportation for tunneling, if you were in running sands, this could be used to help with that tunneling activity. The reason we want to do EICP biocemented soil columns is that we have some advantages over the traditional techniques like soil mixing or rigid inclusions. One advantage is that the EICP solution is near the viscosity of water. So with that low viscosity solution, it takes low energy and small equipment for installation. Also, this is an in situ treatment method, so we have minimal spoils. You wouldn't have to deal with spoils taking out off-site and landfilling them. You would have little to no impact to the existing built environment. So if there's adjacent infrastructure or you need to treat soil under existing infrastructure, that would not be a problem. And potentially we're more environmentally friendly than Portland cement stabilization techniques, as Dr. Kavazianjian spoke about briefly. So the objective of my research program was to scale up the application of EICP for biocemented soil columns at field scale with conventional grouting equipment. So at the CBBG, we have a focus on commercializing products, and we feel that it's important that we use conventional grouting equipment because our ground contractors already have that equipment. So if they don't have to make that initial capital purchase for new equipment, it makes it easier for them to adopt our technology. Also, we wanted to show we can evaluate the effectiveness of EICP biocementation at field scale through non-destructive and destructive testing. So as Paula mentioned, we have the CBBG Soils Field Laboratory in Mesa, Arizona. This is where I did my field scale testing. Here is the pictures of our test pit facility. Here is a schematic showing the dimensions, about 12.5 feet deep, and holds almost 100 cubic yards of soil. So here we are filling the pit. We used a locally available washed quarry sand. This is a permeation grouting technique, so we need to be in clean sands. We offloaded the soil at the ground level with our front end loader, and then we manually spread it in the pit with no compactive effort. Also here are our injection tubes, which we actually wished into place just because we had low confining pressure in the pit. So here's our testing program. We did 7 different columns. We had column diameters ranging from 0.3 meters up to 1 meter, column heights from 1 meter to 2.4 meters. We mostly used an injection rate of 2.1 liters per minute. That's on the low end of permeation grouting injection rates. We did that because we had low confining pressure. When we did the 1 meter diameter, we amped that up to 8 liters per minute, which is getting closer to what you would normally use in permeation grouting. We embedded moisture content sensors in columns 1 and columns 4. We did CPT testing in columns 1 and columns 2. We did downhole seismic on columns 3 and 5, and we did a pile load test on column 5. So here's a schematic of the injection. We use a tube and manchet or sleeve port pipe. This has different injection ports that we isolate with a packer to do the injection. Here is our peristaltic pump that we used, our buckets full of solution, and our packer going down into our TAM into the ground. So with our embedded volumetric water content sensors, we use Teros 12 sensors, and we varied the placement radially and with depth. So in column 1, this is a 0.3 meter diameter column. We put 3 sensors in the design treatment zone. We put 1 sensor at the edge of the treatment zone and 1 sensor outside of the treatment zone. Column 4 is a 0.44 meter design diameter. We put 2 sensors within the treatment zone, 1 sensor at the edge of the treatment zone, and 1 sensor underneath the column. So here are our results. We actually did 3 cycles of treatment in all of the columns with the same treatment concentrations. So for column 1, you can see the spikes. This is when we did our injections. The sensors that are inside the treatment zone, which are sensors 1, 2, and 4, basically received the solution when we injected it. But when we get to sensor 4, which was outside of the treatment zone, it really didn't receive the solution at the same time it was injected at that specific depth. So that's telling us our injection is getting where we want it, not getting outside of it. We do see the sensors outside of the treatment zone get solution, but that's later on and due to drainage, most likely. In column 4, we had our sensor right at the edge. Sensor 4 was at the edge of the treatment zone, and sensor 3 was at that same level. And what we saw there is that at that larger diameter column, we did get the injection solution at the diameter we wanted, but we got less of it. It didn't quite reach that same peak. So that was a good lesson for us to learn from these embedded volumetric water content sensors. Next, we did column penetrometer testing. We had 1 control test, 3 CPTs penetrating the columns, 1 in column 1 and 2 in column 2, and then 2 outside the outer edge and just outside the diameter in column 2. And here is a picture of the exhumed column. You can see the CPT trace of where the CPT penetrated the column. So our CPT results were that 1, our tip resistance in this green line here is our control, wasn't really discernible whether we were in cemented material or not. So that was unfortunate. We feel it's likely because we were in low confining at relatively low cementation levels. So likely the cementation was cracking as the CPT tip went down. And also we could have had a deflection of the CPT because it's a relatively small diameter. However, our sleeve friction and friction ratio did clearly pick up when we were in the biocemented zone. So we're hopeful in the future that could be used as more quantitative. Here it's just it's more qualitative that we were an indicator that we're in the cemented zone. We did estimate our relative density based on the CPT results in the control sounding, and we estimate that is 32%, so relatively loose soil. We then did downhole seismic velocities to get the compression and shear wave velocities of our treated soil. We did this with the purpose-built downhole geophone setup that ConeTech had built for us. We put that down the TAM, the injection tube, and then we made waves on the surface and measured the downhole wave velocity. So for the compression wave, we used a plate. For the shear wave, we did polarized shear waves, and we used a steel box tube for that case. What we found was column 3 was a good column to test because we had uncemented soil at the surface and then only the bottom half was treated in that case. We found the compressive wave velocity increased from 600 to almost 1,000 meters per second in column 3 going from the uncemented to cemented zone. And the shear wave velocity, columns 3 and columns 5 both peaked out around 2 meters. We had 280 meters per second, almost 400 meters per second shear wave velocities in those columns. And we correlated that with shear wave velocity calculated from the control sounding, which we calculated a shear wave velocity at that depth of about 128 meters per second. So it's a very nice improvement. We then did load test, the biocemented, we did a pile load test on column 5, and then we did plate load tests in the pit on uncemented soil. We did plate load tests at the surface, and then also we embedded the plate 1 meter into the soil and tested that. We found that the biocemented soil column increased the capacity 3 times over the superficial plate and 40% over the embedded plate. For the stiffness modulus, we had similar results, 3 times increase over the superficial plate and 35% increase over the embedded plate. Then we excavated the soil and revealed our columns, and that was obviously a very exciting day. So we found that our dimensions were as expected. Our length dimensions were very good. We were a little bit smaller on diameter in some cases, a little bit larger in others. Column 7, that large diameter column, had the biggest negative % difference. When we went to that large injection or higher injection rate, we actually got solution up the annulus of the column, even up to the surface. So we think that has something to do with why we didn't get our full diameter. Here you can see the 1 meter diameter. Here is a 0.3 meter diameter column. Here we are taking down the 1 meter diameter column. It was quite a big task to cut this down and take it out. And here is column 3. I just wanted to show you this is what we typically saw, where we had a clear designation between the treated zone and the untreated zone below, going from cemented to uncemented soil. We then did needle penetrometer testing. We had previously correlated needle penetrometer with biocemented soils in the lab, and we found that readings over 200 newtons correlated with an unconfined compressive strength over 500 kPa. The target strength for our tests here were 500 kPa, and we had 90% of our readings over 200 newtons. So that was exciting. Column 7 gave us a chance to test the strength, the needle penetrometer readings with distance away from the TAM. And with that, we found that up to 2 meters away, we were clearly over the 200 Newton threshold. At up to 0.3 meters away, we were kind of hovering around that 200 Newton zone. These readings were actually taken below the treatment zone in the residual biocemented area. Then we tested carbonate content. There was not a clear pattern with depth. That tells us that our TAM system to inject the soil at intervals with depth was a sufficient system. We did see decrease in carbonate content with radial distance, going back to those embedded volumetric water content sensors that showed we weren't getting the full solution out to the outer edges. That makes sense and indicates that with higher injection rates, we can probably even those carbonate contents up with distance. So in column 7, up to 0.2 meters away, we had a less decrease in carbonate content, and so we're hopeful with that higher injection rate we can achieve more uniformity. We then tested unconfined compressive strength in the lab. We subsampled specimens from the base of column 2. We had, on average, we were within 20% of our target UCS, which was 500 kPa, and we had an average carbonate content of these samples of 2.3%, so we feel it's representative of the other columns in the pit as well. So the conclusions from this testing was that we were able to complete our 7 field scale biocemented soil columns with a diameter up to 1 meter. We were able to show we could increase the downhole seismic by 100 meters per second or more for the shear wave velocity. We had the column load test showing us that we did increase the strength and stiffness. The exhumation after testing showed the column dimensions were similar to our target dimensions. Our calcium carbonate did decrease with radial distance, but again, we're hopeful we'll get less decrease with higher injection rates. And lab testing on our subsampled specimens show that our strength was near our target. Our future work is wanting to implement this in a natural field setting. We want to test this out with more variable soil conditions and also with higher overburden pressure, again, so we can increase those injection rates. We want to advance the verification of our downhole seismic method. We think that's a relatively easy and promising method, non-intrusive method to verify our results in the field. We want to measure the long-term durability of biocementation from EICP treatment, hopefully in a field setting, a natural setting. We want to implement EICP treatment in saturated conditions. This would allow us to validate this as a method to prevent triggering of liquefaction. And lastly, we want to conduct a fate and transport analysis on byproducts of EICP. As Dr. Kavzianjian had mentioned, ammonium is a byproduct, and we want to be able to extract that in a most efficient way. And that's it. Thank you.
Okay. Thank you. Thank you, Dr. Martin.